Today we're going to do a quick video on how to use Mastrings to investigate antibiotic resistance. This is a really quick and easy experiment, so let's get started. Today I'm going to be using two different bacteria. I've got E. coli and I've got Rhodospirillum rubrum. I recommend having two different ones because it's really great to be able to compare samples. You're going to need your nutrient agar plates and then a set of Mastrings, and we'll talk about those in a second. So the very first step is to take your samples, and I'm just going to take 3 drops and transfer them onto my agar plate. I'm going to take my L-shaped spreader from the sterile bag, and I'm going to use it to push my sample all the way around the surface. You want to get right into all those edges and nooks and crannies. This technique is called creating a lawn. The last step is we're going to take our mastering and place it onto the surface. You can see that I'm putting the mastering face down and gently pressing each of those points onto the surface of the plate. Lid on. We'll do the same for the next sample. Now we've got both of our samples plated up. We're going to incubate them for 24 hours at 37 degrees C, or you can even do them for 48 hours at room temperature, and we'll see the results in a couple of days. We've waited 48 hours and these are our results. I really like using Rhodospirillum rubrum because check out that colour, it's pretty cool. What you can see around some of the dots is a zone of inhibition, so an area where there isn't any growth of bacteria. In this case, S, that's streptomycin. So you can see that the Rhodospirillum rubrum is really susceptible to streptomycin. And in fact, you can get your ruler out and go ahead and measure it, and measure all of these dots and compare the zone of inhibition at each antibiotic. You can do the same for the E. coli and chart all of your results and compare how these two different bacteria perform against different types of antibiotics. It's really as simple as that, so I hope you have a go at this, and thanks for watching.